I want to thank the organizers and ILSI for allowing me to speak. I hope that if you attended this morning's session on Cyclospora, you're not too offended by seeing some of the same types of themes, but they're inescapable when we talk about foodborne parasites, especially when we talk about Cyclospora and what we've been going through outbreak investigations over the last 2 years. And I suspect we'll be visited by Cyclospora again in the near future. When we first started to talk about putting together a presentation on technologies for prevention and inactivation, and knowing about what we're used to, particularly with Cyclospora, because that's been the focus of the FDA and CFSAN over the last couple of, I want to say, decades, longer than I can remember. I decided that maybe there should be a little bit more of a change in tact, and it's only because, first of all, I think Ynes covered it very well, and the other thing is there's only a limited amount of information that we can talk about the technologies to inactivate or to prevent the parasite. But what I thought we'd do is give you just a little bit of insight about the strategies, the limitations, and the prevention and the detection of it, to start fostering some ideas of maybe there are better ways to look at this as opposed to just trying to figure out how to find this in the environment. We know a lot so far generally about these types of organisms, but there's got to be a better way that we can approach this. And so I came up with the title of Insight, Strategies, and Limitations, but then I thought, well, I had it a little bit backwards, and so maybe we should talk about limitations first after we talk about the insights. So let me just set the table for you. What are we going to talk about when we look at the insights in these types of parasites? And I'll be very frank with you, I'm only going to brush by the helminths because I think we can get a lot more out of what we know right now about the coccidia, like Cyclospora, Cryptosporidium, and I'll even touch on a little bit on Giardia, but not much. So let's define the problem. We're going to look at the perceptions and the realities, because a lot of times the public will hear there's a parasite outbreak, and they think this big worm that's going to be attacking them, and they just miss the nuances of what a foodborne and a waterborne parasite can actually be. Or they think about their dog or their pet and roundworm and heartworm and so forth. And then, just like Ynes, we're going to look at some of the numbers from the same source, so I won't spend much time on it. I think the bulk of what I'd like to do, though, is talk about the challenges and the limitations, and there are many. And that will probably be the bulk of it, because there are a lot of things that we need to know. However, there are things that we just don't. And maybe that's where we need to focus a little bit more on. And then from what we can glean from those limitations, I honestly believe that you can develop strategies from what you don't know. And I know that sounds a little counterintuitive, but I'm going to try to make you see where I'm coming from with that. So in those strategies, it's a matter of what can we do with what we know, and perspective. Can we find solutions from a different point of view? So first, a little bit of quick hits about parasites. Biologically, naturally, food and waterborne parasites are broadly classified. You heard Ynes touch on the protozoa and the helminths, which are commonly referred to as the worms. These include tapeworms, flatworms, roundworms, and the like. She also used the same type of terminology of the neglected diseases. And we don't hear a great deal about these. When somebody is suffering from some type of intestinal distress, the first thing is it's either a virus or it's a bacteriological type of event. Very rarely do you go to a GP, present symptoms, and they immediately begin to suspect that you have a parasite. Time goes by. When it's not resolved by any type of antibiotic, then you begin to look and look for OMP or ova and parasites. So time goes by. It's not something that we are really trained for to be in the forefront of our minds. As you saw from Ynes's slides, a lot of these parasites come with very complicated life cycles. Some we know, like toxo. Some we don't know as much as we really should. So you may think that Cyclospora's life cycle is very, very simple. There's the environment, there are humans. And there is a mature stage that you need to, for the parasite to become infective. As hard as we've looked, we can't find it in the environment, and we can't identify other animal reservoirs other than humans. Have we looked far enough or long enough? And we really can't answer that. It may be just as simple as that. There are also pre-patent periods, the time between infectivity and the time the patient begins to present with those symptoms, overt symptoms. That complicates things. And if you were in this morning's session, it particularly complicates things when you're trying to do epidemiology of an outbreak that's associated with either a waterborne or a foodborne contamination event. Because these parasites do not present like bacteria or viruses, which can be relatively quick. The onset of cyclospora. The pre-patent period is from 7 to 14 days. Trying to do any type of trace back and really trying to find the source is very difficult, particularly when you're talking about an organism that is transmitted through produce, because that produce is no longer there. Short shelf lifetimes. And lastly, when you talk about parasites, particularly the coccidia, there really is a significant disparity in the availability of analytical methods. Because of what we don't know, it makes it very hard to develop those types of analytical techniques, particularly those that can be used in a regulatory situation. One of the things that Ynes pointed out, and that I'll mention at least one or two more times, is the fact that with a lot of these organisms, there is no vehicle to enrich these parasites. Enriching is a great vehicle for doing molecular or microbiology, particularly with bacteria. Put it in enrichment media, grow it up, and you increase your opportunity and your chance to detect it and identify it. Have a suspected cyclospora outbreak, and it's what you have is what you got. The only things that you really can do is do large sample volumes, large sample masses, large sample volumes, and then try to concentrate any washes or water that you have. That's your best shot. And then doing that creates other complications that make detection that much harder, whether you're trying to identify it by traditional microscopy or you're using molecular techniques. The last one I think a lot of people will agree with. We don't have that here mindset. We do. We see it all the time. These organisms are out there. It may not be considered endemic in the United States, particularly with cyclospora. Cryptosporidium, though, is here, has caused significant levels of outbreaks of illness. We see cyclospora constantly. Whether it's larger outbreaks or smaller outbreaks, it's there. All right. And there are factors which influence that. Now, Ynes did also point out, and she was actively involved in the microbial risk assessment for foodborne parasites. And I just want to present you with some understanding of the risks that risk factors that were put into play in trying to rank globally those parasites which affect the food supply throughout the world. Naturally, public health is a concern, and under public health we talk about total global illness, that it's attributed to a variety of different parasites, both protozoa and helminths, that include mortality, morbidity, persistence of chronic infections, persistence of acute infections, and mortality. Then we're going to talk about the microbial ecology. What is the global distribution of these organisms? Are they widespread? Are they localized? And then, of course, how does it impact animal health? And when we talk about foodborne outbreaks, we have to be careful that we do not just become myopic about human food, but pet food as well. And so we've taken to making sure that when we talk about the food enterprise at the FDA, we talk about food and feed, because they also impact us. And that's kind of the overlap that we may have between the FDA and the USDA. We certainly have to consider animal health in all of this. The last two you certainly can't leave out in this day and age anyway, and that has to do with the global economy. When we talk about agribusiness and trade, and the social and economic impacts. And also, intertwined with this has to be human behavior. So this committee listed and used those factors and came up with the worldwide top 24. And here they are, and attached to them you'll see that they also have next to them the most significant food that is usually found to be contaminated by these particular organisms. Naturally, helminths such as tinea, top of the list. How does this list really kind of reflect what we may be usually accustomed to? It's quite different, as Ynes showed you in the 2011 paper, which I'll get to in just a little bit. Let's still stay globally. Let's talk a little bit about what are the significance of some of these parasites worldwide. Well, in Europe, there's about 2,500 people who are usually affected by foodborne parasites. Trichinella, Echinococcus, different than what we may experience here in the United States. In Asia, really, there are no national data. This goes to the effect that a lot of times we don't pay attention to these types of diseases. Or they're endemic, and there are a lot of asymptomatic carriers that may be present in those areas of the world. Same goes for African nations. In the United States, we do have some susceptibility to tinea infections that do cause neurocysticercosis. But it's the last one, and I apologize for those people who want to listen to about worms, but it's the last ones with the protozoa that I think we should focus on, because I think this is where we are more susceptible, primarily because of what we don't know. Parasitic protozoa are increasingly being recognized in the United States, and I highlighted and waterborne. I know as we go to the next couple of slides, whoops, let me go back, that we talk about Cyclospora being a foodborne organism. But because of the linkage that you usually find in the commodities that are identified with illness, it's hard to believe that water is not involved. And I say that more easily now only because there's no evidence to the contrary. There's no evidence at all, but I think it's safe to say that it's a good bet. I put this slide up here just to show some of the food categories that were also investigated in the worldwide risk rankings. But I do want to just focus only on these areas, the aquatic animals, the dairy products, and plants, principally because these are where we're going to find principally outbreaks with, say, crypto or cyclospora. So when you talk about shellfish, for example, as Ynes pointed out, a lot of them are filter feeders. They are perfect to concentrate some of these organisms in contaminated waters. When we talk about plants, fruits and berries, well, that's what we've learned so far that Cyclospora likes to get to us through. We became very much aware of the outbreaks of Cyclospora in the mid-1990s because of the imported raspberries from Guatemala. So obviously these types of things that have a close association with the environment are more susceptible to being carriers and contaminating foods as we eat them. There was a study that was done in 2013 from colleagues in Canada that actually looked at the prevalence of some of these food and waterborne protozoa, principally Cyclospora, Giardia, and Cryptosporidium. A lot of the fresh leafy green commodities that were collected and analyzed at retail showed these types of frequencies of both presence of Cyclospora, Cryptosporidium, and Giardia. It's interesting to note, however, where a lot of these produce came from. In the study that was purchased at retail, 93% of those retail products that were surveyed and sampled came from the United States. There's no information on where they came before then. And that's where things become very complicated. And this is not to shirk the responsibility that we may be endemic for these types of parasites. But the things that complicate investigations like this is just because a finished product comes from a particular area, the components of that finished product may come from somewhere else. And traceback investigations are complicated by those facts. And I know you can't read the other side, but I won't test you at the end. This is, again, what Ynes showed in the Scalen paper in 2011 that looked at the incidence of foodborne parasites in the United States. It was a follow-up of the 1999 Mead paper that looked at bacteria, viruses, parasites, and their relationship to foodborne illness and foodborne death and the economic impacts that come from it. I won't spend really any time on it, just to reiterate the impact that even these parasites at low levels of infectivity can have on economies and public health. I would say that even though these numbers are low, I want to point out that these numbers really may not be true indicators of the incidence of illness caused by them. I'm not to say that they are 100 times greater than this, but I'm sure enough definitely able to say that these numbers are much lower because of the fact of the reporting scheme for these types of parasites. Cyclospora now is a reportable disease, or a reportable illness, but it's on a voluntary basis. Is that correct, Ynes? I'm out of my depth there, but I just wanted to point that out. Before it was not. And that's only once an individual goes to the physician. So these numbers are really rather low. So let's move from these insights and let's talk a little bit about limitations. Identification, prevention, and control are really dependent on these factors. We have to understand the parasite. We have to understand human behavior that's associated with those factors that the parasite may be connected to. And we have to recognize the impact of agricultural and retail practices, the role of farm to table, and recognizing the impact of the globalization of food trade. Why are these important? If you don't know much about the parasite, you're not going to know where to go to look for it. If you don't know much about the hosts, you won't be able to look at mitigating properties. Understanding human behavior. This goes to the whole spectrum from people who work on the farms, people who pack, people who distribute, people who stock the shelves, people who buy. It'd be an interesting exercise to go into a grocery store, go into the produce aisle and just watch somebody. Watch their habits, the picking, the selecting, and it doesn't take much to think about where were their hands. Now, Cyclospora may not be a human-to-human contact, and so that last part, human-to-human type of transmission, traditional fecal-oral route, because of what is needed for it to become infective. But that's not to say that that same type of concept doesn't go on earlier on in the process from farm to market. What are the behavioral practices of individuals? And it's a fair question to ask. We also, as consumers, as Ynes pointed out, we have started to migrate to exotic foods, bear, boar. These are things that also contribute to our practices. What about the consumption of our practice of some suspected consumption practices? How many people like raw oysters? Okay. Me too. Other practices, where there is no kill step. These are also human behaviors. Sushi, if you want to talk about Helminths, well, sushi is depending on the source and the quality of the fish, are a perfect vehicle for anistichiasis. Human behaviors. I think how well we understand these factors really dictates how successful we're going to be in finding mitigations to this, whether we know fully everything about these types of organisms or not. All right. What about, I think we need to understand why we need to consider these. If you talk about the environment, we're not just talking about our environment here, but the location of the farms. What's in proximity to those farms? Is there farm security? All right. What about runoff, agricultural concerns? I don't know if we could ever modify human behavior. I will never stop eating raw oysters. All right. But you can certainly take into consideration those types of things. Laboratory analytical capabilities are a big limitation. If you don't know much about the biology of the organism, if you don't know much about the genome, if you're hampered by the availability of those resources, advances are very, very slow. This is just a total drawing to give you an idea of the complexity that you may need to understand to try to understand any particular parasite or pathogen. When we talk about harvest times, right? A lot of hand contact. I don't know if anybody can really pick it out, but in the upper right-hand corner, those are harvesters picking up dropped apples, right? Which really is a no-no. All right. Rinse tanks. Environmental factors. Farming practices. How do you irrigate? Do you filter your water? What you eat? How you do your business? Where? And what are your personal hygiene practices? And then lastly, diagnostics. And I think you understand when we particularly talk about cyclospora, what this implies. That describes the diagnostic capabilities in a nutshell. I do believe, though, that that optimism really is that sometimes you can find solutions from the most complex types of scenarios. And I think it's really just a matter of your perspective, how you look at it. So when you look at these types of things, you can end up asking questions. First understanding, though, what are we talking about when we talk about mitigation strategies? Mitigation strategies, risk management, inactivation, preventive controls, these are all just synonymous with trying to stop the transmission in the environment to food through your table. You have to understand that if any solution you come up with, can it be scalable? Can you bring that laboratory solution, that idea, to the field? And will it be practical? And then what do you need to overcome any laboratory methodologies? And do you really have to detect the parasite? So in the FDA right now, we are continuing doing some research to overcome some of the gaps. It's very hard for us to detect the organism on fresh produce, because by the time we get something that may be contaminated, that may not be the same produce that's associated with the outbreak. The other deficiency that we have that we're trying to overcome is how do we sample large volumes of water? Clean water is easy. Dirty water is not. And although it may not sound like a sexy research project, it has long-ranging impacts to be able to do that. I know everybody has heard about whole genome sequencing. Cyclospora is no exception. And trying to work out the whole genome for an organism like this would be very beneficial. How we go about that is the hard part. And it all starts with having the resources necessary to do it. And cyclospora, in our case, is very hard to find. So I'm going to leave you with this last thought about being able to find the parasite. We're always looking for the parasite after the fact, at least when we talk about foodborne contamination. And this applies both to protozoa or the helminths. Can we try to do it beforehand? And that is, can we do more environmental surveillance? We don't even have to find the parasite there, but can we put into place an effective means to prevent? And I think that focus will start principally with water. And like I said a little bit earlier, at least for Cyclospora, it may not be just the water, but I think in the environment, looking at agricultural factors and the like will be the way to start looking. And perhaps implementing those types of environmental proactive preventive controls would be a good way to attack, attack some of these organisms that may be in the environment and affect produce, which really does not have a kill step. It's ready to eat. And with that, I'll just end. So thank you.